if there, I don't see any other hands up or questions. So with that, I'd like to thank Elena for a wonderful presentation. And now we can go to Dr. Al George and hear his presentation.
Great. Thank you, Tim. Can you see my slides okay?
Yes.
All right.
Yes.
Great. Well, listen, it's a pleasure to be here. I want to thank Nanion for setting up the webinar and for inviting me to present some of our data on using the Synchro patch for recording from human cardiomyocytes. I also want to thank Elena for quite an illuminating talk, if I may just add a pun. Anyway, so we are an early adopter of the Synchro patch technology. We've actually recently benefited from the Nanion Loaner program, and shown here in the right lower quarter is our newest addition to the laboratory. So I'm going to talk about human-induced pluripotent stem cell-derived cardiomyocytes. Most people, I think, are familiar with this technology, for which the Nobel Prize was awarded several years ago to Professor Yamanaka. This is a technique where you can take a cell from an individual and reprogram it to be a pluripotent stem cell, and then from those stem cells you can generate any other potential differentiated cell in the body. And for my talk today, we're going to focus on making cardiomyocytes. And why do that? This has been really an area of intense investigation over the past few years. I think most people have used this technology to model genetic diseases of the heart, and others have used it to investigate drug-induced toxicities, including proarrhythmic screening of drugs for safety pharmacology. And there's been some progress in using them for drug discovery and target validation. We've also used this to really explore the molecular pharmacology of some novel sodium channel blockers, and that's going to be something I'm going to talk about today. Our legacy Synchro patch, which we purchased and installed back in October of 2014, so nearly almost 9 years ago, has been a workhorse for us. This has been really transformative technology for our laboratory, and we're pleased to see that the progress in developing this technology has also advanced. I'm going to highlight some studies that were done on this legacy platform that are listed here. All of these have been published. The first study will be an example of how one might use induced pluripotent stem cell-derived cardiomyocytes for looking at novel pharmacological agents, in this case, novel sodium channel blockers. And then I'll highlight some collaborative work that we've done and contributed to that highlights some of the genetic risk factors for cardiac arrhythmias and how we've ascertained their molecular physiology. So this first paper was led by Franck Potit, who has since left my lab and is now a scientist at Icogen. This also represents important and strategic collaboration with Paul Burridge in my department, who is an expert at the cell biology of iPS cells and cardiomyocytes. This is our first paper studying novel sodium channel blockers on human cardiomyocytes. Here is simply describing for you some of the details of that study as it relates to the automated patch clamp. Here are the two internal and external solutions. I want to thank Elena for going over in detail the technology. I won't repeat that. We used, for this study, we wanted to focus on sodium channels. So we used an extracellular calcium channel blocker and a cilodipine and a cesium-based internal solution to prevent potassium conductances. And here are sort of the metrics of how this study was done. We used a high density of cardiomyocytes at that time. These are single hole, low resistance chips, and some of the other metrics that you can see there. And we had to optimize the conditions, but at the time of this study, we were able to get an average of almost 60% cell capture in each 384 well plate, with really good average seal resistances. And overall, the success rate all across the entire plate of measuring robust sodium currents sufficient for doing pharmacological studies was about a quarter of the 384 well plate. So given that we only capture about 60% of the cells, most or half of the remaining cells were really robust. Over time, we've been able to improve on this, but everything really depends on cell culture for this type of cell. And here's just some examples from that paper. I won't go into detail, but we were able to look at tonic block of peak sodium current and use-dependent block, and used, in this case, 5 different drugs and compare them across several concentrations. This work would have taken a long time to do by manual patch clamp, and there's no way we'd have been able to gather the robust data sets that we were to do this work. So just an example, we looked at lidocaine, ranolazine, lacosamide, which is known to be a unique sodium channel blocker for the treatment of epilepsy, and then 2 investigational compounds, one named GS967, which was a very potent blocker of persistent sodium current, and it was almost selective. It had very little effects on peak current, and then a derivative of GS967, GS6615, which at the time was being developed for clinical use in cardiac arrhythmias. I think it didn't make it quite out of clinical trials. Both of these drugs were investigational at the time. No one had tested them on human cardiomyocytes. So we were able to derive really beautiful tonic block experiments and separately use-dependent block. Most of these agents have local anesthetic-like effects, therefore they have use-dependent block. But we were really surprised to see that GS967, which was thought to be primarily a late sodium current blocker, had profound and high affinity use-dependent block of the human sodium channel, which no doubt may contribute to its potential efficacy. And beyond that, we were actually able to derive kinetics of block for all of these drugs and measuring on and off kinetics, or on and off rates. Again, something that would have been extremely labor and time intensive to do by manual patch clamp. So this I first show as an example of the ability of using automated patch clamp on human cardiomyocytes to do detailed molecular pharmacology. Pivoting from that, I wanted to share with you some collaborative work that we did with a group in the Netherlands led by Caroline Remy and Connie Bazina and her team. And this was to explore the involvement of microtubule mechanisms to traffic the cardiac sodium channel to the plasma membrane. In this first paper, the study was done to explore the interaction of specific microtubule proteins, in this case, the N-binding proteins, EB1 in particular in this study, and to look at whether that's involved in the trafficking of the cardiac sodium channel NAV 1.5 to the plasma membrane. Our involvement was to look at cells for which this protein EB1 was knocked out. And these, again, are human IPS cell-derived cardiomyocytes for which one can do knockout studies, relatively straightforward. And we compared the sodium channel current density at the membrane, comparing the control cardiomyocytes with these heterozygous and homozygous EB1 knockouts. I think these are the homozygous cells. And there was a profound difference. And this aligned with the data from Carol Ann's lab, showing that EB1 and microtubule assemblies are essential for trafficking the sodium channel to the plasma membrane. Now, EB1 was quite interesting, but the story got more interesting when Connie Bazina's led international effort to do genome-wide association studies in the arrhythmia risk gene syndrome called Brugada syndrome, identified another member of this N-binding protein family, a gene called MAPRE2, which encodes N-binding protein number 2. And of course, the logical thing was to decide whether or not this too had a role in trafficking sodium channels to the plasma membrane. So again, we turned to the IPS Cell technology platform. Paul Burridge's lab did a knockout of MAPRE2. Again, this is a homozygous knockout. And then we measured sodium current in cardiomyocytes differentiated from the control IPS cells or the MAPRE2 knockout. And again, similar to the EB1 story, we see that there's a reduction in sodium current density, which was inferred to suggest there was impaired trafficking to the plasma membrane. There was no voltage-dependent differences between the sodium channels recorded in those two cell lines. There was no kinetic differences. It was really just current density magnitude. Now, at this point, again, all this work has been done on our original SynchroPatch 768 platform. Going forward, because we were doing so much work with cardiomyocytes, it just behooved us to really move toward current clamp, which the legacy system that we purchased was not enabled. And so, fortunately, skip that. Fortunately, Nanion, through their loaner program, let us borrow a new SynchroPatch 384 system, which was placed in our lab in April of 2021. Over the ensuing several months, we were able to gather sufficient preliminary data, both on cardiomyocytes and some other cell types, that we were able to compete successfully for an NIH shared instrument grant, which was funded earlier this year, and we bought the instrument outright. And happy to say that this is really a win for us and presumably for Nanion, showing the value of this loaner program. So now what we've done with that is we've been able to pivot now to measuring not just sodium currents and other things under voltage clamp conditions, but we've been able to begin to optimize use of this cellular platform for measuring action potentials. In order to do that, there's been a tight collaboration between my lab and that of Paul Burridge, and I want to highlight the work of postdoc in Paul's lab, Mallory Blankard, who's worked closely with Carlos Benoit in my lab to really optimize the culture conditions necessary to get robust currents recorded and action potentials recorded on this platform. So this is just a timeline of how the cells are prepared. Paul Burridge's lab has a unique way of introducing a selectable marker for cardiomyocytes. And so once the cells are differentiated and then selected, they then undergo a dual dissociation protocol that we have found was very valuable for preparing cells optimal for SynchroPatch recording. They're first dissociated at 16 days post onset of differentiation. And then we do a second round of differentiation on around day 30, and then the cells can be plated for use on the SynchroPatch sometime later. And here's just some details about the media. Questions about this probably would have to go to Paul Burridge, not necessarily me. Another thing, when we prepare the cells on the day of recording, here is the protocol for that. We clean the cells with calcium and magnesium-free PBS. We digest the cells. These cells are digested with an enzyme called liberase. They're very gently triturated, and then they're suspended again, placed on the cell hotel, and then we let them sit in the cold for about 30 minutes before they're recorded. And so we've been able to optimize a series of recording conditions that we used. We start out with current clamp, and here are the intracellular and extracellular solutions that we use. This is still a fluoride-based solution. We leave out the calcium channel blocker in these experiments because we need the calcium channel to be active in order for there to be a proper action potential morphology. But then once we measure action potentials, we switch from current clamp and pivot to voltage clamp, during which we can then immediately measure sodium currents, and following that, calcium currents, and then we swap out calcium from the extracellular solution with barium, and we measure the calcium current again. Of course, with barium as a charge carrier, one gets larger currents. And so this is sort of our scheme, and this has become really routine in our lab. And here's some examples of action potentials that we recorded on the new SynchroPatch 384. Here's just an average action potential from these cells, which are predominantly ventricular in origin. This is an example of recordings from the same batch of cells, all on the same plate, all within a time span of less than 30 minutes. Action potentials are measured. We pivot to voltage clamp and measure sodium currents, calcium currents with calcium as a charge carrier, and then finally barium as a charge carrier. So a very efficient, fairly high throughput system. Here's just a blow up of the screenshot from the SynchroPatch showing again an average action potential. In addition to the cells we've cultivated in collaboration with Paul Burridge, we also have an ongoing collaboration with Dawood Darbar across town at the University of Illinois and in Chicago. His lab is generating atrial cardiomyocytes, which have a shorter action potential. And here's just a lineup, an average of the two recordings done on the same day on the same plate, showing atrial in blue and ventricular in red, showing the difference in action potential duration. And then some examples of sodium current, which I've already shown you. This will give you an idea of how that looks. And again, here's calcium currents. And so at the end of the day, this is a very efficient system for us right now, really trying to harness this technology now to do a lot of different experiments, some of which are looking at genetic models of human cardiac arrhythmia, genetic cardiac arrhythmias. Others are looking at the pharmacology of channels in these cells. So really great system. Lastly, this is an example of some pharmacological studies that we're doing, and this is sort of a throwback to the original work that we started with to look at the role of microtubules in trafficking the sodium channel to the plasma membrane. Here we actually used a probe compound that it's a dirty drug. I'm not going to make excuses for it, but one of the things it does do is it does modulate microtubule function in a specific way. It has other functions as well, but we looked at this because our collaborators in the Netherlands had demonstrated that, in fact, this compound, which we go by the nickname SB2, does alter the kinetics of insertion of sodium channels into the plasma membrane and potentiates trafficking. And so here we see again data recorded under voltage clamp. Sodium current density is enhanced when cells are pre-treated with the SB2 compound. It doesn't have much of an effect or any effect really on the voltage dependence of activation, or but it does seem to shift in activation kinetics to hyperpolarizing potentials. And then given that it does affect action potentials as well. These are recordings that we couldn't have done before on the original synchro patch, but now we can do those seamlessly, and we're really pleased with that. So just a little show and tell on the kind of things you can do with human cardiomyocytes with automated patch clamp. I want to thank the folks in my lab, particularly Carlos Vanoye, who is right here in the front in the plaid shirt. Franck Bautit, who is now an employee of Icogen, and Mallory Blancard and Paul Burridge's lab really contributed to this work and some of our funding here. And of course, great thanks to Nanion for not only their loaning of the instrument to us, but really more of a collaborative relationship in the way they support us, and really love working with their scientists. So with that, be happy to take a few questions.
Thank you, Al. That was a fantastic talk. I'm scanning through the messages now. Yarmar, do you see any new questions in the chat? Ah, I think I see one. There is one question that just came in. Question is, the double mutant of EB1 and 2 viable, and would there be any sodium current left? I'm not sure I quite understand that.
So if we made a double mutant between EB1 and EB2, I think is the question.
Oh, okay, yeah.
So it turns out there is also EB3. So it's a little bit more complicated to answer that question. I will say that Paul Burridge's lab has made multiplex knockouts, and it does, there is some additive effect. I think those experiments are still incomplete, so I won't make a comment about that. But I do think that there is an additive effect of knocking out multiple end-binding proteins.
Okay. I had just more of a technical question on the experiments where you're measuring the current density. Were those all done on single hole chips? Do you know?
Those are all done with single hole chips. Okay. Yes.
And I was wondering if you had considered using multi-hole chips. That way you might get a cleaner separation because you're able to average across more cells in each well.
That's a good point. I think in the beginning we probably gave some thought to that. The reason we probably haven't done it is already the cell density required is pretty high, and the making the cells is quite a time and labor intensive process. And so to make actually more cells, we'd have to have a really good justification for it. So I don't think we've tried to use multi-hole for cardiomyocytes. And
Okay, that's a
have
good
used
point.
multi-hole for many, many different cell types.
Mhm.
And, in some cases, it really does even out the well-to-well variability, which is really valuable for doing pharmacological studies, and also for boosting current amplitudes in cell lines or in situations where the current amplitude is small. So we definitely use them. We haven't tried them yet with cardiomyocytes.
That's a good point. Yeah, the cardiomyocytes are a precious commodity. That's true. Okay, I think we have another question that just came in. It says, "Thank you for the nice talk," and it went away. Just a sec. "Thank you for the nice talk. How problematic is the injection of holding current leading to shortened action potentials, in your opinion?"
So, the cardiomyocytes derived from iPS cells tend to have a shorter action potential.
Mhm.
And that's probably related to the fact that they are immature cardiomyocytes, and the amount of calcium channel expression is not optimal to have a long, sustained, depolarized plateau phase. Um, More recently, and I didn't have the data in the final form to share today, but the older the cells get, there's a tendency for the action potentials to get longer. And
Okay.
so I think it's a product of immaturity mostly. And I think that we've also toyed around with the idea of cheating a little bit, and instead of recording in the presence of extracellular calcium, maybe record the action potentials in the presence of extracellular barium to kind of boost the calcium conductance, or at least conductance through the calcium channel, to see if that would help. That's very preliminary, and I think we need to work a little more on that. But I think the main thing is the cells are at day 30, 32 that we've been studying them. They're still pretty immature, and lately we've been going a lot longer, and the action potentials probably are going to look a little more physiologic.
I see. Okay, there's another question, which is it possible to control the resting membrane potential and measure spontaneous activity in current clamp mode?"
Um, Yes, it is. And I didn't point that out, but in the cardiomyocytes, we don't see a lot of spontaneous activity. Um, if we force the holding potential to -80, we still don't see a lot. And I think it's primarily because these cells are acutely dissociated, and cardiomyocytes that are beating spontaneously in culture are in syncytial layers, and they're attached to one another. And you get, I think, once you dissociate them and uh, disperse them, they lose that ability, sadly. And we
Mhm.
haven't figured out a way to kind of get it back. We've tried all kinds of things, including bombarding them with isoproterenol to see if we can't activate them and have more spontaneity, but they don't. But that said, if we hold them and then induce action potentials, we get pretty reliable action potentials to fire. So that's one of the limitations. We can't see spontaneous action potentials. For that, we turn to the cardioexcite system, which we also have,
Uh-huh.
and look at monolayers of cardiomyocytes, and they give beautiful spontaneous activity. So
Uh-huh. That's a that's a good point. Yeah, the dissociation is something that makes a big difference, can make a big difference. And the cardiomyocyte or the cardioexcite or the even the flex site
right.
or
So there is another question from Ali,
Mhm.
who asked me if we use the same holding current in all the wells, or is it individualized? It is.
Mhm.
It is the same holding current for all the wells. I don't think we have a way to individualize it at this point.
That's true, but I think that's coming. That's
That's
something
coming.
that the engineers and software guys are working on. Yeah.
Right. Yeah.
Um, there's a question about intracellular calcium. This is great talk. I just wanted to ask about intracellular calcium transients. Have these been characterized in these cells, particularly because the calcium currents seem to exhibit very small inactivation?
Yes, we've actually measured intracellular calcium transients on a 384 well platform sold by a company called Vala Biosciences, using basically calcium sensitive dyes. And they are very robust calcium transients. I'm not sure what the question was, but they're very robust intracellular calcium transients that do mirror the time course of the action potentials. And again, those calcium transients are really variable depending on the state of maturation. So again, Paul Burridge's lab has spent a lot of time and effort to optimize maturation of the cardiomyocytes, and looking at intracellular calcium transients has been one of the readouts for that. So.
I've got a couple of technical questions I think you could probably knock out real fast. One is, what's the temperature that you record at? And the other is, what's the maximum days in culture that you've gone to?
Yeah, we've been doing still everything mostly at room temperature with the new synchro patch, which has temperature control. We at least aren't beholden to the building's temperature. We've actually locked it in at 22 degrees Celsius. So that's a little more day-to-day control. We have the option, obviously, of heating the cells now. And that's something we haven't done systematically yet, but we're exploring doing that. Obviously, recording at near physiological temperatures would perhaps give us more insight into how these cells are really behaving. The other question was about the oldest that we've studied. Yeah, I'd say the most, we've probably taken it out to between 40 and 50 days, probably not quite to 50.
Okay.
That's the oldest we've done. So they're still healthy at that point, so we could presumably go farther, but perhaps we're too impatient.
Yeah, that's understandable. I think there's a few questions about potassium channels. And so one of these questions asks, it states, "Wonderful talk, exclamation point. Have your lab tested to record IKR and IKS in iPSC cardiomyocytes?"
Yes, certainly we have. We are able to see outward currents in these cells, and we try to differentiate them pharmacologically by using specific blockers. So for IKS, we used a compound JNJ303, which in vitro is really high, has very high potency for blocking recombinant IKS. We do not see much effect on the cardiomyocyte outward current, very small amount. And then IKR, we used E4031 as the pharmacological probe. There again, we see some block, but most of the outward current is probably not either IKR or IKS in these immature cardiomyocytes. You can ask me what it is, but I can't tell you what it is. I don't think we have figured that out yet. It's probably a mixture of things, but we have not done those experiments on the older cells, and so that should be done.
Yeah,
haven't done that yet.
Okay, let's see. I think there was several questions along the lines of what you just answered, and you've already answered about individual eye hold. Does anybody have any additional questions in the audience, or because if not, I think we've gotten through the
Marcus
chat.
Stand up.
Oh, Marcus.
Hi, Al. Very impressive, fantastic talk. Thank you so much. I have a question. Have you ever seen any pharmacological difference in the channels when you look at them in iPSCs versus heterologous expression systems?
Yeah, so I think there are differences. If we looked at recombinant NAV 1.5, for example, in hex cells, the voltage dependence of inactivation is very shifted toward hyperpolarized potentials compared to native cardiomyocytes, which is published, and also with the IPS cell-derived cardiomyocytes. So it's a little different. Kinetics of inactivation look about the same. I think the activation curve is about the same as well. So there are some differences. What those differences are due to, I couldn't begin to explain, but obviously there are a lot of differences between hex cells and cardiomyocytes.
Thanks.
And we haven't done, I don't think we've done a direct comparison of calcium currents in the cells yet. We do have recombinant CAV 1.2 and CAV 1.3 recordings that we've done systematically, and we just haven't made the comparison with the cardiomyocyte calcium current. That's a good thing. We should probably do that.
Okay. Well, Thank you, Al, and thanks everyone for the participation in this discussion.